So welcome to everyone, and thanks ever so much for coming. I'm an interesting person to have in a series like this. I'm a web biologist. I was a computer programmer, and I got bored of being a computer programmer, so I went and did a biology degree. But then I spent years running a wholly wet genetics and cell biology lab with no maths at all. And then somehow it turned back on about 10 years ago. And this is maybe because of the questions I was asking, or it may be just that it was always living there. But more than half of everything I've done since has been based around working with mathematicians and modelers, more recently actually computer scientists and deep learners. I'm going to give you a talk which doesn't have very much hard maths in. I'm going to explain what it is we did, but mostly I'm going to talk about why biologists need mathematicians, why mathematicians should be really interested in biology, and how you would go about joining the two of them. So that's the question we're interested in. We want to know how cells move around. And one of the things you can immediately see is that while there are lots of ways you can simplify this, cells are extremely complicated. Quite a lot of the movies I will show will be time-lapsed, so they'll be sped up a lot. But these actually look like this. If you look at them down the microscope, you can see them migrating and running around. So it's very fashionable. The past 10 years has shown an unbelievable increase in the power of microscopes. As the microscope's better, so the computers control of them and the cameras and everything. So we have humongous quantities of data. And with data comes understanding. But cell migration was always important. Why was it important? Well, imagine this is a problem that we in the world of biology spend lots of time worrying about. You started as that. You ended up as something a bit like that. I'll tell you two stories, just because I can. I had test tube babies. We got too old becoming professors, and so we needed a bit of help. And I wanted to see my babies when they were fertilized embryos. And they said, no, absolutely not, you can't do that, it's illegal and so forth. That try didn't work. The next try, I said, I am a professional cell biologist and microscopist, but I'm really not sure that the kit you've got there is ideal. And they had my babies out like whoosh, and so I got to see at least one and probably both of my daughters as 4-cell embryos looking just like this. And this is, of course, I'm not showing pictures of other people's kids. This is a GAN-generated baby and not a baby that ever actually existed. Anyhow, two anecdotes to sell over wheels. So you can't just turn to one of these by going. It's not going to work. Well, it'll be balls. You can't go from one to the other by just changing the identity or the shape or what have you of the cells. We'd be balls with eyes and hair and so on and so forth, but we'd still be. What has to happen to go between these two is all the different cells have to go in groups and layers, and they have to migrate against one another. And that's why you are the lovely complex shape you are. I'm not going to go into the details, but you need cells to move. And so before you're born, you need cells to move, but at every time afterwards. So if you work out in the gym and your muscles get bigger, the way that you get more blood vessels to your bigger muscles is that the cells that line the blood vessels move. They migrate and they create it all on their own. They don't need any particular external stimulus. They organize themselves into new blood vessels by moving. If you cut yourself, I do a lot of gardening, so I cut myself an awful lot. Then you put nasty things, bacteria and so forth, in your bloodstream. I'll show you a movie of this in a bit, but the only reason that bacteria you get in your bloodstream don't kill you is that you have cells moving to try and catch them. You think about your brain And you've all, I'm sure, seen enough images of what a brain looks like in terms of complexity of cells. What happens is you get layers of nerves, and then one layer of nerve migrates out connections, which are just like moving cells, and then migrate and they find the right thing to connect with in the next layer. So the nerves from your eyes can migrate down the optic nerve to connect with the brain. If you can't move, then no nervous system. And then the thing that paid me for 15 plus years before I came to UCL is that all this cell movement, of course, also causes bad things. So if you get arthritis, it's because cells from your bloodstream have moved into your joints when they shouldn't. And if you get cancer, and it spreads because the cells decide to move. If the cells don't decide to move in cancer, then it's a lump, it's no problem. But if the cells do decide to move, then you could die. So cell migration is something that you ought to be worried about an awful lot, etc., etc., etc. I mean, I just give you a few. Right. When we try and study cell migration, we have a hard time. Why do we have a hard time? Because you saw the cells are moving like mad and they're changing shape and they're moving to different places and they're responding to things and so forth. They're very fast and complicated. We can get a huge volume of data. Each frame from one of my microscopes is between 4 and 20 megabytes. I can make one of the microscopes take one of those every minute, going all the way overnight. I can make it do it at 20 different positions in a sample. We can absolutely fill the world with data, but what we want to do is understand the data, and that is the problem. Different cell types, again, you know this, but actually you have to talk to the right kind of biologist to get a reflection on it. Different cell types are very, very different from one another. One of your skin cells and one of your blood cells, they've got the same machinery, but they're using it in a completely different way to do a completely different thing. And we want to understand both, but you have to know about all these differences. And then there's the fashion. And the fashion is actually going to be a main theme of what I talk about today, because medics in particular love determinism. When you go to the medic, you want the medic to say, you are sick because you have pyelonephritis. And it doesn't matter if that doesn't mean anything, it doesn't matter if the medic isn't certain, that's what people want. And biologists work in the same sort of way. They want a very deterministic story. They want to know, you did that because you were hungry, or you wanted to meet someone or eat someone or whatever. And actually, you of all people know everything's random. And the whole of life is based around randomness. And then out of that randomness, we try and take a little bit of order. And biology is very, very good at doing that. But even so, the way you should be understanding things is order being yanked out of randomness, rather than by saying, you did this because that. That's not really how things work. And the other thing, and I'll come to this again and again through this talk, this is really important. When you are training mathematicians, all the way through undergraduate, all the way through a PhD, every seminar, you are used to the language of maths and the way it's done. But the language of the biomedical field is verbal. So even though they're describing stuff often that is very, very, very mathematical, medics and biologists would love to say the cell wants to do this. And of course the cell doesn't want to do anything, it's a cell, that's stupid. But it's a very effective way to describe things, but it's a very non-mathematical way of describing things. And that's the reason that I've ended up doing a lot more, working with a lot of mathematicians and doing a lot of math computing related things. I haven't counted this recently, it's probably lots more. There are loads of papers modeling particular aspects of biology. more than 200 about modelling individual cells migrating. Most of them no one much reads. Some of them are very influential indeed. Hopefully you'll have a little bit of understanding why after this. The data glut is still going up exponentially. We are just installing a new microscope in CDB, which will generate terabytes of data a day. Wherever you look, there's tons of data. So mathematicians and computer scientists working with biologists is actually the only hope for moving forward. We can't keep using words to describe things and getting orders of magnitude bigger data sets and then just trying to look at them and say, okay, the cell wants to, it can't work. So there we are, there's an exaltation of you are what we need. Now this is the thing I'd really like to understand. This is called chemotaxis. In chemotaxis, cells orient themselves towards something that is a diffusible stimulus. So in this case, that is a bacterium. If you have them in your bloodstream, they will kill you very, very quickly. You'll get sepsis. That is a neutrophil. A neutrophil is a kind of white blood cell that guards against bacteria. This is just blood. You can tell it's blood because look, those are red blood cells that are starting to get a bit sickly. And you see the neutrophil chases and chases and chases the bacterium until it catches it and kills it. And that's why you survive if you scratch yourself or prick yourself with a rose thorn. Rose thorns are disgusting. If you ever look at a rose thorn in an electron microscope, they're this festering zoo of microorganisms all on the end of a sharp thing. And if you prick yourself with that, you're injecting a whole load of bacteria that grow every 30 minutes. Your mathematicians can work out that exponential, think where that leaves you in a couple of days. So you've got to have a good way of dealing with it, and it's chemotaxis. So we want to understand how the neutrophil knows where the signal is, but actually, verbally, that's fairly easy. Mathematically, maybe less so. We want to know how, when they get that information, they can actually physically move. This structure at the front is called a pseudopod. We want to know how they make one of those. So, if you asked 100 biologists now how that neutrophil moves and catches that bacterium, they would tell you a story that I'm going to tell now. It's cockamamie, it's rubbish. I have to say so clearly this is a straw man, but it helps for me to explain the straw man, to explain why we do what we do. So if you have a little bit more of an attractive chemical here, and a little bit less here, it might only be, cells are very, very small, so it might only be 1 or 2% difference across the cell. Then the mainstream idea is that the cell has a sort of compass and it works out what the direction is. So the cells detect chemicals using things called receptors, which are molecules that sit on their cell surface. And the outside part sticks to something the cell's interested in, and the inside part sticks into the cell and communicates the information. Ever so important to think about that, because no information really can get from the outside into a cell normally, unless the cell builds a machine to do it. So the idea is that the cell says, okay, we've got more receptors turned on there, and we've got slightly less receptors turned on there, and we're going to calculate it somehow. And then when they've calculated it, the idea is that the cell says, okay, I should be going this way, sticks out a new pseudopod, and that's how it steers. In other words, the signal creates the pseudopod, and this, and it's nice, it's very, very deterministic. It's very verbal, it's just the sort of thing that biologists like, but it's nonsense. Why do we think it in the first place? This is an essay I did, the last thing I did before I I got too busy running my own lab. Just now the kids have gone and I've moved to UCL and I've started doing these experiments again. When you have a micropipette, and the micropipette is full of an attractive chemical, you get this unbelievable response. I mean, this is the cell's chemotaxis. Now, what you see, you won't take the first one. When it moves here, you see that the cell sticks out a pseudopod. When it repeats, I'll show you again. So people concluded from this experiment that the cell is sitting there, looking for gradients, but comparing sensitivity all over its surface. So ignore the first move. Right. Come up here. Come up here. Right. So you can find instances where the cell seems to know. Now, the way we, the biologists, think about this is just a change in direction. But if you were to consider points on the surface of the cell and consider the diffusion of a molecule out of the tip of that pseudopod, out of that pipette, and so forth, then you'd realize there's no such thing as a simple spatial stimulus. That's daft. What happens when you move this pipette is you get humongous changes all the way around the cell. So you can't really tell anything from this experiment alone. You have to go and analyze it. That's the reason that people think this. One of my first students who did this sort of work, she was doing a PhD in the computing department, and she got really sick of it because it was not rigorous and came to do biology. I've never had a score as good as that since. But she came and looked. So when you have a cell that has moved towards the micropipette being there, and then you move the micropipette up here, and you follow the tracks of this cell, you see that actually it doesn't make new pseudopods in order to steer towards the new direction. Actually, they're nearly always wrong. That's not the straw man I showed you is simply not what's happening. This image that the PhD student from the computing department who came to work with me did is such a beautiful and evocative image that it actually won a bunch of prizes as an art in science. And the PhD student is now a professional artist in the US, works with a mathematician who does lovely Turing patterns and things, and they create works of art. So the theme of jumping fields is quite common. Okay, here's an interesting thing you see. Look at this pseudopod. You can see that this is how the cell is moving. This is at the front. And what happens is it splits in two. And then it splits in two again. And then this one splits in two. And then occasionally, ones like this, the cell gets sick of them and takes them away. So the cell isn't saying, okay, I need to change direction. I'm going to stick out the pseudopod in this way. You've got this self-sustaining pattern here. Again, an audience like you will recognize pretty quickly what's going on here is maths. There's some kind of Turing system, there's some kind of instability. It's self-fueled growth leading to instability. And that's what determines how the cell goes. We think that the cell moves around autonomously by just organizing its components. We think it's autocatalytic because anywhere where you have a pseudopod, that makes more pseudopod. I'm going to express this in an equation in a moment. And so what you have to do is you have to understand these rather mathematical things called pseudopods, which move the cell around, rather than, I think, this verbal argument that says the cell wants to go in that direction with the highest attract. Above all, the conclusion is that the pseudopods, the way the cells move, are feedback loops. It's not a thing, it's not an object, it's just the manifestation of some maths. So we did this nice work where we looked at all the pseudopods that the cell randomly makes. And we looked at the ones that the cell keeps. And we looked at the ones that the cell retracts. And you can see that these are doing a really bad job of steering the cell in a good direction, which should be straight up here. But the cell is keeping the ones that are in a good direction much more often than it's retracting them. So if you're going in this direction, for example, you're about as likely to be kept and retracted, whereas if you're in the wrong direction here, you would be kept and you will be retracted. So the cell's randomly generating things that will move it around, and then if they're in the right direction, then it might keep them, and if they're not in the right direction, then it won't keep them. And I like this for lots of reasons. It's easy to reconcile this with information theory, because each pseudopod is spending a long time growing and integrating information and so forth. Whereas the decision to say we're going to go left or right is an instantaneous decision. It's quite hard to see how you accumulate information, for example. And I'm trying to do a project on this with UCL maths as we speak. So if you want to summarize this, the biology zeitgeist is like this nice man at the end of the Charge and Light Brigade, where the man on the horse says, charge, and everyone else behind them goes, charge, and they all follow this one guy. And what you will find if you look at biology is biologists spend lots of time trying to find the man with the hat. And the conclusion that we make is that biology actually looks like a termite man. And if you expected to find one termite at the tips of those things, you would be stupid, because that's not what's happening. What's happening is the termites all running around, interacting with one another, and then it gets biased a bit by the sunlight and their gas and so forth. And that's the idea we should be following. So we don't want these pathways that you will see if you ever look at biology. What we want is something that is random and biasable. There's a great list of phenomena that people try and explain. I'm not going to do those today. I'm just going to show you a little bit of modelling and how well we can do it. So, in order to model those cells that I've shown you before, we just model the edge. And it turns out later on that actually just modeling the edge will do fine. So you can model the cell as a perimeter in 2D space, and actually you can model it rather effectively doing that. This is a bit of a surprise because cells are full of stuff and they have a nucleus in and so on and so forth. But whatever the explanation of why they're moving, just considering the edge turns out, as it happens, to be sufficient. So if you have 100 or 400 points equally spaced along the perimeter of the cell, and then you move each of them subject to a set of equations, then you'll get an evolved interface, and that's fairly nice and easy. And what we say is some number of points are active. We keep a parameter for each point, which is the activity. But the activity in most of the points we consider to be 0. And a few of them we consider to be higher. And that's going to give you a pseudopod, just like I showed you, and the rest of the cell isn't a pseudopod. So if you are active, then you move out from the perimeter, normal to the edge, and we measure that just to check. It's an important thing to check, but it's true. And then everyone else gets sort of pulled along behind. So you model the connection between all the points as a spring with a certain amount of resistance so that it doesn't change too fast. So far, pretty easy. But it turns out that if you go modeling this, it's a disaster if you just model a bunch of points connected by springs. Why is it a disaster? Because saying the cell sticks out something here, then the points are going to rapidly spread out there. And as the rest of it contracts behind them, the points are going to come back together. And fairly soon, your model is going to break down. You also have a terrible problem that occasionally points will cross over and the perimeter will become twisted, and that crashes the model every time. So you have to do something to make the edge behave well. And the first thing that we did is we mapped our edge onto a level set. And then, having mapped it onto a level set, we regridded equal points along that level set. And that worked, that was lovely. We published a paper showing that, and we could make a very nice simulation. But the computation required was enormous. We're quite lucky because the guy who did this for me, I hired from the University of Strathclyde, and he was both very, very greedy and very, very sociable. And so he talked to all the faculty in the University of Strathclyde and borrowed their computers. And by borrowing their computers, he went in and put a little Unix routine on them that said he could have it when they went using it. And then there was one year, there was a very long Christmas break, so we had all the computers in the whole of the Strathclyde maths department, and that was where in those days we got enough computing power to do this. It turns out that you can do incredibly better in this kind of model by using a parameterized finite element method. And if you are interested in that sort of thing, I can, you can read these papers, or I can point you towards the papers showing it. But it uses about 1% of the computing, and it actually gives you more accurate results, because every time you jump from the edge to a level set, and the level set back to the edge, there's a rounding error. Whereas we found that the finite element, parameterized finite element method didn't. So that's how we moved the edge. We then used a system that had been developed by a fantastic mathematician who was a follower of Alan Turing. If you are aware of the history of all of this. Alan Turing, as well as breaking codes and inventing the first computer, wrote the first paper on the mathematics of evolving biological shapes. That was an incredible paper. Wonderful man. Anyhow, so the activator that is moving the cell forwards is being synthesized in proportion to the square of the amount of activator over 1 plus activator squared, so it runs from about 0 to 1. So it's being created in response to itself. And then you have two inhibitors, one of which is a local inhibitor, which just is being made locally in response to the amount of the activation diffuses away, and another global one. And if you don't have a global inhibitor, the system works to make shape, but the shapes it makes are permanent. So if you have a random edge moving a little bit, making things a little bit, and anytime one bit sticks out a little bit further, then the A term is autocatalytic. So the moment noise brings one point a little bit above its surroundings, it will immediately start growing and inhibiting all of its neighbours. But the trouble with all of that, actually, sorry, I'm saying it wrong. I'm saying it in the wrong order. Forgive me. The trouble with all of that is the moment the cell has gone away from being circular and got polarity, then all that you have is the polarity self-sustaining itself. So you also have a local inhibitor that says if you get too active, if A becomes too high in one region, then it destabilizes it. And there's all sorts of biological trouble with this model, like this integral here, the loop integral. You can't have that information. You can't know what the concentration of some substance is all the way around a cell, because the information you can't get from the edge of the cell to the central point. This has caused the world lots of fun. Anyhow, Matt Nielsen, who is the person from Strathclyde Maths who did it, working with John Mackenzie, who was the head of Strathclyde Maths until recently, solved this using a surface finite element method, all of the terms were arbitrary, which is to say, it's a toy model. A doesn't represent anything beyond a behavior, but it works. And we got this. And this is the sort of magic of emergent properties, because I've explained to you totally, I'm giving you a complete explanation of what we're doing here, but I bet you wouldn't have expected it to come out looking exactly like a moving cell. You see the pseudopod sticks out, and then it becomes unstable and splits in two, and then one of them grows and one of them retracts, and the one that grows becomes unstable and splits in two, and so forth. So you get this path, slightly jaggy path, as it splits the pseudopods and then chooses one, splits the pseudopod and chooses one, and so on and so forth. But actually this remains, it's more than 10 years old this now, and it remains the most reflective, I'm trying to use the word right, it's the most good-looking model of a cell movement. Hard to say accurate because the terms are arbitrary, but it's the most behaviorally similar, let's say. Doesn't turn, so that's completely random, there's nothing steering that. So we added a term. This is to do with the receptors I told you about. So what this is about the receptors saturating. So as more and more of your receptors are activated, this term grows slower and slower, which is real. With some randomness, actually this is the old formulation, this is a naughty formulation. We had to do a much more tight formulation, randomness and so forth. But the point is, in the next bit, we didn't do anything clever. We didn't add any of the things that biologists have emerged. We just said, wherever the signal is higher, then the coefficient of positive feedback at the activator is a little bit higher. We made it in proportion to the difference, so the difference is tiny, but if you have two autocatalytic systems in competition with one another, you only need to give an infinitesimal boost to one of them, and it will win. It will outgrow the other one, and that's what happens. So now we model this cell moving around, and in a moment I'm going to put a gradient on it. Start now. That's the gradient. And what you see is by miles the closest thing to what real biological cells do. You remember that arty picture I showed you? It kept going for a while, and then it slowly turned around. Still makes mistakes, because it's not a determined system, it's a stochastic system, but with strong bias. And this set of partial differential equations has given us the most beautiful simulation of a human taxon cell. And we're very pleased indeed. The paper describing this has been cited about 200 times. It's a well-loved paper. I think 200 is the number. I haven't looked for it. It's influential within a narrow group. We didn't manage to convince all the biologists about this. Why did this wonderful thing not convince all the biologists? Because even though it describes everything really nicely, the biologists wanted each term of the equation to be something that is tangible. They didn't want me to say there is an A term and the cell moves out according to the A term. They wanted to know the name of the molecule and the difference between the molecule turned on and the molecule turned off. And we don't have that, no one has that. So this has been influential, but not general. Do you see this lovely thing, the models here, despite totally emergent property, despite nothing of the salt being coded in, this model beautifully recapitulates the observation that the active bit sticking out will be kept if it's going in the right direction, and it will be retracted if it's going in the wrong direction. Fantastic this, because we did nothing to hard code that in, it just emerged from the feedback. So we explained all of the things we wanted to explain in cell migration. with actually a really simple four entity model. And there's lots of other things to show that one should be confident in this, like as you add more and more noise to it, this is almost no noise, coming to a lot of noise. It's very, very robust. Because it's a stochastic model with strong bias based on positive feedback, so it can cope with noise. It gets less good if the noise gets more, but it copes with it. So that's what you need to do, your cells need to do that. So it's robust, it's broad, it's conceptually simple, it can replicate all the things we wanted, and it agrees with, fortunately, me when I say that the cell is a bunch of stochastic protrusions, and you select them all. Here was a really funny thing. Matt, who did this, we got very, very interested in what would happen if the cell that we simulated got caught up in a really unfriendly environment and had to squeeze its way out. So Matt modeled a whole load of solid objects, immovable, and ran a simulation of it. And to our absolute amazement, when the simulation hit these things, under appropriate conditions, it ate them. And this is called phagocytosis, and this is how cells eat things. If you have one of your immune cells and there's yeast in your bloodstream, then what the immune cell does to the yeast is exactly what is shown here. And he was talking about this result in a conference in, I can't remember where it was now, Germany, I think. And the American who spoke straight after him had been measuring all this stuff and finding exactly the same parameters. So we wrote a lovely paper together saying that actually phagocytosis was an emergent property exactly the same way that migration is. The problem with this is the thing I'm saying, that medics biologists are terribly grounded in the tangible. We want to know what is the molecule, we want to know how does it turn off, how does the cell break it. If you can't say what each of A, B, C, and sigma are, you can't put a name to it, then you are very much limiting your ability to speak to medics and biologists. And I'll say this one more time, explain more. It's very, very important never to show an equation when you're talking to medics and biologists. Okay. I'm going a little bit slowly, but I'll skip some things if it doesn't work. So, if you have cancer, there's this term called metastasis, which is the cancer spreading, and the cancer spreads by cell migration. And actually, deaths are pretty much all caused by metastasis, cancer spreading, and as the cancer cells spread, they end up in the wrong place and they get in the way of things. So they block nerves and they block blood vessels and so on and so forth. And metastasis is a kind of cell movement. So if you look at melanoma, how many of you know what a melanoma is? About half. So a melanoma is the nastiest kind of skin cancer. It's a cancer of your pigment cells. And when I say it's nasty, what do I mean? I mean that if you have a melanoma, there's a high probability that the cells will spread from the melanoma to other parts of your body. So this cell migration and the cancer being nasty, it's actually the same thing. So you can see, looking at these melanomas, that they're not just spreading randomly. Because if they were spreading randomly, the shape of a melanoma would be Gaussian. It started presumably about there. And if the cells were just moving around and migrating randomly, then you'd have lots more cells there, and as they randomly migrated out, you'd have a perfect Gaussian modulated by things getting in the way. But what you actually see is that there are bits that stick right out, and if you ask your doctor, is this a melanoma, and should I be worried, the doctor will look at it and say, does it look like a Gaussian, or does it have sharp edges and unevennesses? And if it looks like Gaussian, the doctor will say no. And if it has asymmetry and things and differences over its surface and sharp edges, then they'll be very worried. So we say it's probably chemotaxing, it's probably spreading to something. So this is the sort of thing we do in the wet lab. We made a device, we put some melanoma cells here, we put some serum here, and they spread. They migrate down this little channel that we've given them. And tell you one thing about this movie, it's really frightening. I mean, it's pretty, isn't it? It's just cells moving. But if you had a melanoma, in what's the time? Time's up at the top. In 48 hours, the cells from that melanoma are moving from the place where you put them to a place that's about 600 microns away in 2 days. And 600 microns is the biggest distance between any point in an actual melanoma on you and your blood system. And what we've put in here is serum, which is a blood product. And so what you're seeing here is why a melanoma kills you. The melanoma cells migrate from where they are in the tumour to your bloodstream. And we know the chemical that they're doing it towards. But you can see a very interesting thing. If they were just moving up this gradient, then they ought to move consistently all the way. In fact, they ought to move a little bit faster at this end because the concentration is lower, and I showed you the saturation equation. But what they're actually doing is moving in this tight wave. And everyone behind this tight wave is not directed at all. I'd like you to think about the math of that while I show the next slide or two. Right. So the melanoma spreads out because of chemotaxis, because there's some soluble chemical in you, and the melanoma cells are migrating towards up gradients of that soluble chemical. So you have to ask the question, what's the information that makes a melanoma go? Why does the body make a gradient of something in order for the melanoma to migrate out and kill it? That's stupid, right? And the answer is your body isn't stupid, it's the melanoma that makes the gradient. And the best way of showing this, which surprised the heck out of us, so I'm telling this story out of order for effect, is if you put serum here and serum here. So there's no gradient, no information. And you watch what happens. It's exactly the same. So what's actually happening here is that these cells are breaking down the attractant. So very soon here there is no attractant, there is high attractant here. You've got a flux of attractant coming this way. And the reason you have this narrow wave is that that narrow wave is sufficient to break down all of the attractant that's diffusing across this way. The reason these aren't going anywhere is there's nothing left. The cells in the wave have destroyed it all. So you'll see the familiarity here. It's driven by positive feedback, it's dynamic, it's robust, it's multi-scale. So you're not going to measure it very easily, and it's going to be very interesting to mathematicians. Why can't you measure it? Because it is so evanescent, it's all based on diffusion. The cells are breaking down the attractant and the cells are responding to it. If I take my device apart and go and try and move bits with a pipette, it's gone. So here's some other examples of the same things going on. These are a kind of amoebas we sometimes work on called pictostelium. And they're making a signal and they're breaking down the signal and they're responding to it. And you get this lovely wave pattern coming out. Oh, well, never mind. I'm running out of time. Do any of you know what this is? But let's set it going. This is the exact chemical equivalent. Have any of you come upon this? One or two. This is a Borisov-Sabaczynski reaction, and it's absolutely fantastic. You can do it yourself with a Petri dish with a few fairly normal chemicals, and exactly the same thing is happening. Whenever you get one of these centers standing up, the center is catalyzing a wave that spreads outwards. So the information is spreading outwards in response to diffusion from some original point there. And it was only shown last year, 2023, that your white blood cells do exactly the same. So here's an experiment where some bits of yeast were put in those four corners, and human neutrophils taken from the bloodstream of a donor shortly before were put on there, and you have a thing that shows when they're signaling, and you get these amazing waves of response coming out in response to those yeasts. But you see, you have something that is mad to the biology. The people who wrote this paper haven't even noticed how mad what they're saying is, because all of these cells are chemotaxing towards the yeast. As the wave goes out, the cells chemotax in towards the yeast. Here you see it blown up a little bit, and you see the wave goes out, and as the wave goes out, the cells migrate in, okay? So they're all coming in, but none of the cells are chemotaxing towards a molecule that's been made by the yeast. Because the yeast is just there and it's not letting anything diffuse. The molecules are all being made by the blood cells while they move. It's a positive feedback loop. It's cool. So, when you have an immune response, it's a collective thing. All your immune cells are communicating with one another with molecules like this, and that's how they're doing. So the only way to understand it is through quantitative models. If you go and look in biology, you'll find lots of people using ODE models. They're great. They're instructive, they're very comprehensible, but they won't explain any of the things that I'm showing you there because they're varying time, space, and other things behind. So ODE models often miss the biology. Partial differential equations, stochastic differential equations, work really nicely, but they can be frightening to many. So we use them a lot, but you have to get quite expert in explaining what they mean. And in particular, you must make sure that if you use such models, that their terms and parameters are understandable. Cellular automata are brilliant, but medics hate them because they can't understand how they work. And agent-based models, biologists love, but biologists love them because they're naughty. You can tell your agents to do really non-mathematical things. You can tell your agents to have to make decisions where they cannot, in reality, have enough information to make those decisions. So they're very good, but one must be careful with them. That's a cellular POX model that absolutely perfectly reproduces the cell. Amazing, that one. We don't really know why it does so well at the moment. I've talked much more slowly than usual. No, well, never mind, let's.
I think you have plenty of time because we've got run booked quite a while. Actually.
Okay, right, good.
And it's Wednesday, so I don't think anybody's booked.
Okay, good. Well, okay. I'll make sure I'm finished by about 10 or 12 past. So this is a simple kind of model we use. This is a toy model that we love to explain this process to biologists, so medics. So each cell is an agent. Each cell is following really simple rules in this case, and the red stuff that we are showing here is a diffusing attractant. And this is a lovely explanation of what's going on in all these movies I've shown you. There's the wave of cells. They're breaking down the diffusible, they're creating this gradient here, and then they're reading it and migrating along. And you can model it more accurately by making each agent into something that moves randomly, and then has a bias according to how steeply the gradient goes across it. So this is a proper, well-behaved, parameterized mathematical model, and it beautifully recapitulates what the cells are doing. So this is the sort of model that we favour the best as a way of joining medicine and mathematics. It's a hybrid model. We have agents, so it's agent-based, and the agents are cells, and it has layers that are PDE, showing things diffusing and breaking down and so forth. And we found that by going to that type of model, we can explain more or less anything we wish to in a way that the people that we wish to explain it to can follow. So we can explain why you get a wave, because the cells in the front go a bit more slowly, and the cells behind them go a bit faster, so the thing is self-sharpening. And then actually you have probably a Hopf bifurcation somewhere here, because there's some infinitesimal where the wave goes past and the cell gets left behind. I keep asking people to do an analytical solution of that. We have magic here. I won't show that, it's just showing it's real. I won't show that kind of Okay, so here is that same model, but now instead of making cells go in a 1D path, we're making them go into a split path. Okay? And there's one complexity here, which is there is a new source of more attractant here, but there isn't one here. So what you see is the cells make the gradient, they migrate along the gradient, about half of them go each way, but then more and more go along the one with the source of attraction. It's just what you'd expect, although it's all equations and insert. Well, what happens if the blind path gets shorter? Well, then the cells can break down all the attractants in here and it goes out by diffusion before the cells will arrive. So almost no cells go into here, and they all go into here. But this means that cells are doing a remarkable thing in this model. The cells are looking ahead of themselves, they're looking around corners, and they can tell where it is they ought to go without even going there. It seems completely improbable, particularly as you can make it arbitrarily complex. So it is a maze with the parameters tuned so the cells always do the right thing. I mean, it's a bit unrealistic, isn't it? Real cells aren't going to do anything like that. Except that it turns out we have the maths, and everyone in this audience knows if the maths is right, then it's true. So to our slight amazement, when we went and built the maze according to our parameters in reality and put cells in it, the cells do exactly what the model said. It was surprising because you can only build this device if you have the maths, and no one had built the maths before. Isn't that absolutely marvellous? And it's not just marvellous, I mean it's cool, it's sort of model and cell pornography, but it's actually real biology. So if I prick myself with a thorn, like I told you, then my immune cells have to come out of my capillaries, and they have to navigate through the tissues which are complex and find the infection. And they can't just go in a straight line, they obliterate my nerves and whatever was in the way. They have to find their way around. It's exactly the same as shown there. And it's the same with embryonic development and nerves finding one another and so forth. And I already showed you our models are great, but we noticed a problem. Now look, in this one, same thing happens. You have a plug of cells, and then the ones behind have no signal because the ones in the plug have killed it. And when we looked at this thing I've shown you, you get a plug of cells, but the cells behind know what the answer is. So there is actually an error in the model, and the cells are talking to one another. And there was this enzyme called vinyl cyclase, and we have blocked it with caffeine, which works in all the big time experiments we do, but we inferred that it wasn't working at this tiny scale, and we got gene knockouts, because we can do that easily these days. And when you get a gene knockout, it works perfectly. So when there was a difference between the biology and our model, it was the biology that was at fault. And we learned something about biology. And this is the great thing about all of this. The models are now good enough that every time we see a disconnect between the model and reality, then we know that there is something different we have to model, or there's something going wrong with the biology that we didn't understand. So we're learning cool stuff every time we run one of these. We've suddenly jumped from the model that is nice, but sort of academic explanation of things, to this rather beautiful. I'm going to go through this fast. Okay. the last movie, and then I'll give one or two summary slides. Do you know this maze, everyone? This is Hampton Court maze. This is King Henry VIII maze, and the cells do it fine. The cells do it much better than people do. You can pay good money to go to Hampton Court and go get lost. You see all these suffering people getting lost because they can't find their way out. The cells don't have any trouble with that. But when it repeats, you'll see an interesting thing, like there's a glitch here. We didn't make the maze quite right, okay? Watch what happens when the cells hit that glitch. They know there's a glitch now. Because they haven't got a map of the maze, what they're doing is working out the environment. And in this environment, there is a glitch there, so the cells can work it out. So the cells can actually sense where their environment is by doing this breaking down the chemoattractants and then following the gradient they've made. So what we did is we made a real big glitch there to test it. We made this maze, and this is actually really upsetting because our model's good, as I've said. So we know what's going on here. Some of the cells go through the shortcut. There they are. And now these cells that have gone through the shortcut are breaking down all the chemoattractants, so there's nothing left coming back here, like the model said. So these cells will never get out of the maze. They will spend the rest of their lives stuck in that maze. But you see the cells, by breaking down a chemoattractant and following the gradient they can get in a purely mathematical way, can understand all the paths around them and where they should go. I love this. Okay. This story goes on and on. I could give the whole talk again, but you wouldn't thank me for it. This was just illustrating what we do. If you wanted to do something like this, if you're a mathematician who wanted to model biology, some medics and some biologists believe in the power of mathematics and models. Find them. They would love to hear from you. They would be so happy. Most, meaning 75%, say, of medics and biologists do not believe in the power of maths and models. They are frightened. They think you're much cleverer than they are, and they don't want to play. So you have to find the restricted number of people who are signed up to it. It's really silly, because biologists are very clever. They just use their tools in a completely different way from the time when they do the first year as undergraduates. Second thing, the worst thing you ever see in mathematicians modeling medicine, modeling biology, is beautiful equations that don't accurately tell you anything about the disease you're studying or the cells you're studying and so forth. The way to do that is to get into the lab. Get someone like me. Actually, there are a decent number in UCL. That's why I've moved here, because I like this sort of thing. There's a decent number of biologists who would be very pleased to welcome mathematicians to the lab and say, this is a microscope, you know, look down this bit, put cells in here, now you've got data. I'm doing that at the moment. I'm taking a summer student who has a math background. I've also got a PhD in maths who's coming to learn to express biology as well. So the time it works is when you split the difference, when the mathematicians are learning something that is mathematically interesting, and the biologists and medics are learning something that's biologically and medically interesting, and then you breed a group of mathematicians who can converse with and excite and so forth medics and biologists, and vice versa. It's not easy. If you just go and get a random biology collaborator, the chances are they won't be interested. And I saw a horrible example of this fairly recently, where a guy who's an immunologist came up and said, oh, we had a model to do this, and they did this model, and I don't really understand it, but it says. You want to avoid stuff like that, like the plague. It's horrible, it's not interesting for you, and it's not getting respect from the biologists. Whereas these mazes and cancer cells moving, everyone understands how cool that is. Just showing I practice what I preach. PhD in pure mathematics, liquid crystal pneumatics, PhD in theoretical physics, working in my lab. He generated some data and decided he liked making models more. He worked in my lab a bit and decided that actually what he really wanted to do was microscopy. Everyone's happy. He generated some data so he understands how it works. He started generating data of his own full time. I said never show an equation for a medic or a biologist. Why is that? Are biologists stupid? No. What happens is that if you do biology school, in the first year of your biology, you do this big verbal explanation thing. And in the second year of your undergrad in biology, you do lots more of this verbal explanation, learn all the different exceptions, work on them, think about that. So by the time you're talking with a lecturer, they've had 1.5 decades of the biological style of doing things without ever looking at an equation. And the way math training works is completely the opposite. Your first year, you get the list of equations, and you do analysis in the first year. You do analysis in the first year, anyhow. So you are absolutely soaked in people sticking out equations and seeing what it is they're trying to do. But the medics and biologists just aren't. So if you show me an equation, I can sit there and I can struggle with it for a bit and I've got it, but you're probably 2 slides ahead by then. So even with me, it's best not to show an equation unless you're careful about showing what it's doing. For most biologists, you'll just scale them off. If you are ever working with biologists, you have to keep asking for explanations. And the trouble with medicine and biology is there are so many things, but if you keep asking, the people you are working with get better at explaining the important things to you, the things you need to know, and you end up with a model that encompasses the things you do need to know and doesn't worry too much about the things that you don't, and you get to understand them. Are mathematicians ignorant? No, you've just spent all those years getting trained not to worry about all the exceptions and list all the things you have to worry about, where the biologist hasn't. But biologists who work with mathematicians and mathematicians who work with biologists split the difference between these two positions, and it's really cool. More friends. If it's not interesting to you, you're not going to do it well. I mean, obviously. But it isn't obvious to a lot of people who are working in the field. If I can't explain why your equation or your model or so forth tells me something cool, then my colleagues aren't going to be interested, and I'm not going to be very happy. But if I show that maze, the solving the maze, then everyone understands what that is, whether you understand it as a system of PDEs or a bunch of cells that want to get out of the clean air. Finally, I'm sorry it's been a long-winded thing, but it's cool. If you were in UCL and you wanted to talk to people, there are quite a lot of them. And I'd be delighted to reply to any email saying, I am interested in such and such a biological process, or applying such and such a type of mathematics to biology or medicine or something like that. There are some examples of people who do this all the time. In maths, Philip, Mo, Karen all work a lot with professional biologists and write papers that medics and biologists think are really important. So talk to them if you're interested. And the Turing now has a full-time biologist in it called Alan Lowe, who is looking at how to do machine learning on these sorts of problems as well. There are many, many, many other people. This is just something I put together at the last minute. But if you're interested in this, get in touch. Get in touch with me or one of these. And that's all. Thank you very much.